the title is Promotion of Alleles by Genome Editing in Livestock Breeding Program. I will also flip this list, so for each lecture you have to sign, so we can prove in Brussels that this was really part of the project. So, John, please, floor is yours.
Thank you, Rino. Hello again. So I'm going to talk about genome editing in the context of quantitative traits in livestock breeding programs. It's a simulation study. Some people think it's not true, that we make too many simplifying assumptions. There are some arguments that could be made that yes, those people are correct. But I think you can also make an argument that our simplifying assumptions enable us to think about how genome editing could be used for quantitative traits in livestock breeding programs. So this doesn't advance. So, why are we doing this? So if we remind ourselves, in the last few years we have had this adoption of genomic selection, and we have had as a consequence a massive growth in the number of animals and their genotypes in our livestock breeding program. So for example, this number is already from 2 years ago, and at that time there were something like 1.2 million animals that have been genotyped in the US dairy industry. So the farmers are using this technology because it pays, it returns a profit to them for investing in genotypes. And for us as researchers, it will enable us to have huge amounts of genomic data that we could use to identify quantitative or variance controlling traits in some way. And we propose this hypothesis that you could layer a very small amount of sequence data on top of that genotype data and create very large data sets with whole genome sequence information. And I talked a lot yesterday about the strategy through which we are doing that. And I also mentioned that we're doing it in a few different populations. We're doing it particularly in a pig population, which has a few 100,000 animals. So this is a kind of our resource, and our resource is not unique. There are lots of people in lots of industries that have resources on this scale. And imagine if we have whole genome sequence information, we can start to also exploit other types of genomic data, similar to what Martin mentioned yesterday. So some of you will be familiar with this project, the functional annotation of animal genomes. It's an animal version of the human ENCODE project. And I think about the ENCODE project as having some assays in some tissues that tell us about whether the sequence is doing something in a particular region of the genome or in a particular tissue at a particular time point. So for example, they have here 200 different cell types or cell lines and tissues. They run different assays on these tissues. So an example assay could be the DNA's hypersensitive site 1 assay. So what that assay does is it walks along the genome, and every time it sees that the genome has open chromatin, it displays that, and when the genome has closed chromatin, it also displays that. And apparently having open chromatin is kind of important because if the genes are to be active in that particular cell type at that particular time point, the chromatin should be open in some way. So with these different assays on these different tissues and time points, you can get pictures like this, which represent the whole genome. So for example, if this was one of the assays that represented open chromatin, you can see where it's blue, for example, the chromatin is open, and where it's not blue, the chromatin is closed in that particular tissue. So if we have whole genome sequence information, we can couple this type of assay and this type of data together with our whole genome sequence information and our regular phenotypes to identify causal variants for quantitative traits in some way. So one of the ways in which we could exploit this type of technology is through genome editing. And genome editing is a very simple idea. You can get your molecular scissors and you can just chop away a little piece of DNA that you don't like, and you can stick in a piece of DNA that you do like. So you can add or delete or replace a single base or a string of bases or a bases in a few different regions of the gene. Like most molecular technology in livestock breeding, it has had impact on simple traits. So if we take, for example, these are two half siblings. Nelore cattle from Brazil. One of them has been edited to carry this myostatin mutation. The other one is the wild type, and you can see a very clear visual difference caused by a single edit at a single region of the genome. This is a particular animal, again from recombinetics, that has been edited for a single gene or a locus that removes the horns. It causes baldness. And we can see that this animal has its horns removed, and we can also see this animal is a very happy animal. If we look here, these are again Chinese pigs that have been edited again to have the myostatin mutation, and if you compare them to a typical pig, these have pretty strongly expressed muscle. So these are all very clearly visually evident examples of genome editing. But there is a problem with that kind of an approach, or that kind of a thinking. So if we think about our livestock breeding goals, so let's talk about traits first of all. So when we went to school, we learned genetics in the following way, that there was a single locus, and you could have 3 different genotypes at that single locus, and that each of those 3 different genotypes would display a very visually different phenotype. So this could be coat coloring cattle, for example. And then maybe when we went to university, we discovered that actually there were more than one gene, there were actually 2 loci, or maybe even 3 loci, and that resulted in slightly more categories of phenotypes. But then when we became real animal breeders and we went out and worked in the real world, we realized that actually traits are controlled by thousands of loci. So a typical trait like milk yield, or fertility, or some muscling trait, or any kind of economically important trait that you think about in the livestock breeding program has a distribution like this. It's kind of continuous, and underneath that distribution at the genetic level are thousands of tiny little variants that each affect the trait in a very small way. So, the genome editors that have been operating to date, they've been mostly thinking about traits like this. But if we are to make genome editing really have a big impact in the longer term in livestock breeding programs, we need to start to think about how it could be used for these types of traits, because these types of traits are mostly what matters economically. So we did a simulation study to kind of quantify some of these things, or get a handle on properties of this. So essentially our objective was to develop a strategy to enable genome editing to be used for quantitative traits in livestock breeding programs. And we define quantitative traits as traits controlled by, for example 10,000 causal variants whose effects are all sampled from a normal distribution, so no big effect QPA. We call this strategy PAGE, which stands in our case for the promotion of alleles by genome editing. And for those that are into molecular biology, we apologize, we didn't know that you guys used PAGE to mean something. So I'm very sorry about that. We didn't know about that because we don't know anything about molecular biology. So why did we call it this title? Well, we wanted to be very distinguish something that we think is quite important. So you could think about genome editing as just going out to another source population, identifying a QTL that source population has, and then bringing that QTL into your existing population. And that's one use of genome editing. But that's not really what breeders do. What breeders do is they accept that there's a lot of genetic variation standing within our population. And what we're trying to do over time is select animals that increase the frequency of these existing favorable alleles. And what we wanted to do was use genome editing to speed up that process, to take existing alleles within our population and just increase their frequency through this genome editing. So that's why we call it promotion of alleles by genome editing. It's promoting alleles that already exist in the population. So to kind of understand this stuff, we needed to quantify, how much genome editing resources would be required to achieve significant increases in genetic gain. So, for example, how many alleles do I need to edit per generation? How many animals would I need to edit per generation to, for example, double the rate of genetic progress? We want to quantify the benefit and the risk. Yes, we will get extra genetic gain. How much would that be? And we want to see, how does this impact, for example, the genetic variance and the inbreeding, and ultimately the long-term response to selection. So we set up simulation, which is again very simple. So we have it has three components. We have an ancestral piece which runs for 100,000 years with some random mutation and recombination rates and random selection using a coalescent model that approximates the historically effective population sizes of cattle. We then said, okay, let's imagine this population ran into or emerged into a recent historical breeding program that was managed for 20 generations by a qualified breeder, and that this breeding program could implement genomic selection. In each generation they would produce 1,000 candidates, 500 of these candidates will be male, 500 female. All of the females will be selected to become mothers in the next generation, and 25 of the males will be selected to become fathers of the next generation. In this piece, we assumed that we had genomic selection with perfect accuracy, and our reason for that was we sort of said, if our GWAS is powerful enough to discover causal variants for quantitative traits, it would have to be driven by an extremely large genomic selection training set, so therefore we could assume that accuracy is close to 1. We then had 20 years of future breeding down here, where the breeder again continued with genomic selection with the same parameters, but the breeder also had the choice to edit some individuals. So the breeder could edit the top 5 males that get selected, top 10 males that get selected, or the bottom 5 males that get selected, or all 25 males that get selected. And the breeder could choose to edit these animals for anything between 0 and 100 edits per side. I want to stress that our trait is controlled by 10,000 causal variants, with the effects of these causal variants being sampled from a normal distribution. So there are no what we would call large effect QTL. Everything is pretty small. Our comparison metrics were the rate of genetic gain, what changes happens in allele frequencies, the number of distinct QTN that get edited, and anything that happens to inbreeding. So this is the primary result in terms of genetic gain. So here I've got my 20 generations of historical breeding. Here I've got my 20 generations of future breeding. Then I've got genetic gain represented from generation, what we call -20, so the first of the historical breeding generations. Or we could have genetic gain represented from generation 0, which is the first of the future breeding generations. But what we see is that genomic selection gives us this nice progressive rate of genetic gain. And then at this point here, the breeder has the option to, in addition to doing genomic selection, they can also do this genome editing. So the 25 sires that get selected would be edited for 20 loci. And if we did that, we basically double the rate of genetic progress. So let's just think about that for a moment. So we're editing the 25 sires that get selected for 20 loci. The trait is controlled by 10,000 loci. So for me, 20 loci is a relatively small amount of loci, but somehow it's having an impact that it is doubling the rate of genetic progress. Another thing that you can see that's happening is that genomic selection is starting to have some asymptotic properties, let's say. It's beginning to asymptote, whereas the genome editing and genomic selection strategy doesn't have the same asymptotic. It's kind of still going on a linear rate. And you'll see why that's the case in the next slide if we look at what happens to the changes in allele frequencies. So here again are our 20 historical generations, our 20 future generations, and here are the average allele frequencies at some point across time. So if we take at the start, if we take the solid lines represent the average allele frequencies of all of the QTN. So the blue line represents changes in allele frequencies when I'm doing genomic selection only. So what we see is that there's a very small change in allele frequencies across our 40 generations. On average, we're going from something like 0.5 to something close to 0.6. Right? If we look at the change in allele frequencies for all QTN, and I'm doing genomic selection plus 20 edits, plus this page strategy, I see a very small change in the allele frequencies. Yes, there's a slightly faster change in allele frequencies by doing this genome editing. But what's perhaps more interesting are the change in allele frequencies for the 20 QTN with the largest effect. So remember, our trait is controlled by 10,000 QTN. The effects are sampled from a normal distribution. But there are 20 QTN that sit at the end of that normal distribution which have a larger effect than the other 9,800 or 980. So if we look at what happens to the change in allele frequencies of those QTN, so so happens that they start off with a low average allele frequency, and genomic selection is able to dramatically increase their frequency up to a point, but at some point it starts to asymptote. And what's happening there is that some of these QTN are being lost from the population before the genomic selection is able to drive them to fixation, and that happens due to linkage drives, essentially where they might be being pulled out of the population by negative alleles that they are being in close physical proximity to on the genome. So there's selection pressure that removes them from the population before we can select upon them. The second reason they could be lost is just due to drift. In livestock populations, the effective population size is very small, so just alleles happen to just disappear from the population due to random events in the way in which genomes are inherited and recombined. In contrast, if we look at what happens in the genome editing approach, so I want to remind you that we do our gene discovery in generation 0. So we can only edit alleles that still happen to exist in the population at this time point. And what happens here is that we're able to get these QTN, and we're able to drive them to fixation within 3 to 4 generations using this genome editing approach. And that's because this genome editing approach treats alleles a little bit like they are independent of all of the rest of the genome. They're not being affected by co-inheritance of other alleles. They're not being affected by drift. You're just simply able to lift them and push them to fixation. And if I just go back to the previous slide, so where we saw this asymptoting of genomic selection, and we don't see the asymptoting of this strategy involving genome editing, but we can explain it with the same phenomenon. So the genomic selection is able to fix about 80% of the alleles, whereas the genome editing is able to fix all of the alleles. And I guess I want to stress that this is an important point. If we think, some people debate about the ethics of genome editing, and if you think about the ethics of breeding as being, the only, let's say, the one way in which you can describe the ethics of breeding is, do I act to take the genetic variation that I have in my existing population and with maximal efficiency turn that into genetic gain? That is a kind of an ethical thing to do. I'm not wasting my natural resource of genetic variation. And if you make that argument and you believe that argument is true, then you would say that a strategy that involves genome editing is a more ethical strategy than a strategy that doesn't involve genome editing.
Excuse me, I didn't get your argument about, like, you're fixing stuff earlier, so why do you not have no asymptote there?
So these are just the top 20 QTN hands, but so what the genome editing does is it comes now and gets the next 20, the next 20, the next 20, right? So it sorts out the first 20 biggest ones, then it comes and sorts the next 20 biggest ones, then the next 20 biggest ones. Does that make sense?
Can you repeat the first reason what you said? You said, just to explain why it goes asymptotically after 0.8.
This one here.
Yes.
So what's happening is, let's say there are 20 big, we're plotting here the allele frequency of the 20.
All or just the 20?
The solid lines are all.
All right.
Right?
Yes.
The dotted lines are the 20 biggest. So the way I would see the blue line representing is that genomic selection has been able to fix 16 of those 20, and 4 got lost from the population. That's the way I would see it. Genome editing has been able to fix all 20. And towards Hans' point, so you'll see on the next slide, let me come back to your point on the next slide because I think it kind of explains. So this shows generations on this axis, and the heat map represents the number of distinct QTN that are being edited at any time point. So what we see is that on average in any given generation we're editing about 30 QTN, right? But over successive generations there's some correlation between the alleles that are being edited. And within sort of 3-4 generations we've completely switched the set of alleles that we're editing. So here we're editing, let's say, the biggest 20, then we're something like the next biggest 20, the next biggest 20, the next, something like that. It goes forward. Which I hope answers your question. But the main message from this slide is that across the 20 generations we edited 314 distinct QTN. So this is around about 30 per generation, distinct QTN. And these 314 QTN explain about 36% of the base population genetic variant. But if you divide this 20 into 314, you get something like 15 QTN, right, per year. So if we would need to set up a discovery process that discovers 15 QTN every year, and if we did that and implement that, we would be able to double the rate of genetic progress under the assumptions of the simulation. So in the end, I want to come back to how would we set up a process that discovers 15 QTN per generation. And I guess, these numbers, this becomes tractable, right? So if I tell you the trait is controlled by 10,000 causal variants, you'll say to me, well, we can never find, using GWAS, 10,000 causal variants. But if I say to you, we just need to find 300, that suddenly becomes a number that I think in the next 5-10 years we could be able to discover. And if you argue that that is incorrect assumption, then I would argue that we need to stop doing molecular biology and animal science, because if we're not able to use all of the molecular biology investment that's being made around the world, if that combined isn't able to discover 15 QTN per generation, then why are we doing it? You can answer that. So, another way we looked at it is, so imagine we have a fixed total amount of editing resources, so we can do 500 edits per generation. Let's say that's our total editing resource for each generation. What would be the most optimal way to distribute that editing resource? So we could edit all the sires equally, so if we have 25 sires, that would be 20 edits per sire. Alternatively, we could take the top 5 sires and edit those for each 100 loci, so that would also be 500 loci. So here's our genomic selection on its own. This is the same figures we've had previously. Here's if we do genomic selection plus genome editing, where we're editing each of the sires equally for 20 alleles. And here's the red line is if I only edit the top 5 sires, but I invest all of my editing resources in those top 5, so I edit them for a lot. And what we see is that instead of getting twice the amount of genetic progress, we're getting 4 times the amount of genetic progress.
And you're not overusing using the top 5 more than the other ones. It's equal use of
equal use in that generation, but you'll see on the next slide. In the next slide is exactly the problem you're alluding to, right? So yes, equal use of these sires. Now, I guess just one thing I forgot to mention, and it doesn't matter whether genome editing doubles the rate of genetic progress or multiplies the rate of genetic progress by 4. And it also doesn't matter whether you believe the assumptions or not. But what I would like you to think about is in terms of step change technologies. So we all have done a lot of work writing papers, etc. And if we take any random paper that we have written and we think about what that paper does to the rate of genetic progress in a practical breeding program, it adds something between 0 and 1% extra genetic gain. If I increase the accuracy by changing from G+ to base B, I get 1% more accuracy, I get 1% more gain. That's a very small benefit, right? I don't know, Hans, if you would agree, or but in all of the papers that I have read, there have only been, let's say, a single paper that really drove a step change in the last 20 years, and that was the genomic selection paper. It doubled the rate of genetic progress achieved by a breeding program. And this technology, this genome editing technology, is of the same scale. It's doubling the rate of genetic progress, which I think is something we need to we need to take very seriously. So now towards Hans's question. So on this axis again we have our 40 generations, and here we've got the inbreeding coefficients. And inbreeding is somehow where it parameterizes your ability to make long-term genetic progress. If you have a lot of inbreeding, you cannot make long-term genetic progress. So what we see is that if we have genomic selection on its own, inbreeding proceeds at this rate. If we have genomic selection plus genome editing, where all of the animals get edited for 20, all of the sires get edited equally, it doesn't change inbreeding, the red line. Inbreeding rate basically stays the same. But if we take, for example, the purple line, where we're compressing the edits onto 5 sires, it has a big impact on inbreeding. It causes a big jump in inbreeding. And what's happening here is that although in the simulations the sires contribute to the next generation equally, it's actually their children that contribute unequally to the subsequent generation. So imagine the son of an animal that's been edited for 100 loci. If the normal distribution of breeding values is here, the sons of the edited guys get pushed out, potentially a standard deviation in front of the rest of the population. And then in the next generation, if you go to select a sire, or the animals that are to become sires, you're going to take the ones that are the sons of the edited guys, which are, instead of being the sons of all 25, they're just the sons of 5 animals, which is what's driving up this inbreeding.
Johnny, sorry, I have a question. Like, how many chromosomes you have?
I can't remember, but it probably was either 10 or 20 or 30 years.
And your QTLs were distributed at random across those chromosomes?
At random, with the restriction that it must have been 10 chromosomes, and each of the chromosomes had to have 1,000 QTL, and we had 10,000 QTL in total.
Because I think you would even have slightly different changes if you are putting like always all QTNs on a single chromosome. That would mean that IBD status of that chromosome would get high, much higher than the others,
Yeah.
isn't it? Like,
Yeah, the other chromosomes are essentially not under selection, and they just bounce around at random, right?
But then if you would just combine like in the first, maybe you're not sorting out like the best 20. Like in the first generation you just put the QTNs on one chromosome, then the next time you go on the other chromosome, then the next on the other.
Yeah, you could do that, yeah.
This then might help on the overall average.
On the inbreeding or on...
Maybe because you let more generations and recombinations.
Yeah, I don't know.
But that would be luxury, right? The issue is we need to get, 10, 20 loci to edit, and it's probably they're going to be spread out evenly across the genome, I would imagine.
No, I agree. I just
Sure.
put the question: is there is difference in those strategies if you are randomly placing, or if you are having strategy across the chromosomes?
So in some sense, the genome editing kind of is...
They're taking it out, so you don't care.
For example, we find out that like the inbreeding depression is not very strong on one chromosome, and we do edits on those chromosomes that don't care if we rise genomic inbreeding on that chromosome.
But the genome editing is sort of independent of genomic inbreeding, because the QTLs are... We're going to inbreed those QTLs, but we're not going to touch the rest of the chromosomes.
No. But
we don't interfere with it.
we start to think that we can like decompose the inbreeding depression to the chromosomes.
Yep.
If this is possible, then we also notice that some chromosomes might contribute to inbreeding depression more than the others. So if you are choosing the QTNs on the chromosome that is bringing a lot to inbreeding depression, you will get higher in genomic inbreeding on that chromosome. And this would... But if you are editing on a chromosome that it's not contributing to inbreeding depression, then your rise in inbreeding would influence so much the population.
So the way I would think about editing is, editing enables me to stop selecting on those QTL. So I don't need to. So if I select on those QTL under normal processes, they will drive up inbreeding in the region around about. right? Because they drag stuff.
Imagine on that chromosome, you concentrate on chromosome, you don't have deleterious effects or whatever on that chromosome. Your all inbreeding average is not equally distributed. Your genomic inbreeding is just much higher on the chromosome you are targeting actually, and you don't have to suffer the problems.
Okay, I can agree with that. So, if the science fiction hasn't been enough for you, we now can go to the next level of science fiction. So who has heard of synthetic gene drives? Martin has heard. Thank you, Martin. I was putting my hand up yesterday too. So, imagine we edit this guy, and we make him homozygous, and then we make this guy to this individual, and we produce this progeny. But this progeny would just be heterozygous for that edit, okay? And if we then put that individual in here, and we make this guy to these guys down here, we will see that, you know, just a very small number of the descendants would carry the edit if we didn't select in some way on the edit. But we could actually have what they call a gene drive mechanism. So we are going to edit this guy, and we're going to insert also the gene drive mechanism. And what that does is here's my edit in the middle, and then either side of the edit I add also this CRISPR-Cas9 machinery. So what I do is I make this guy homozygous, and then if this guy mates to this guy and produces an individual that is normally heterozygous, then the gene drive mechanism activates. It cuts the alternate strand and it drives itself in so the individual becomes homozygous, right? So you just need to inherit one copy of this edit from your parent and you will automatically become homozygous. And if you do that then without any selection, you know, all descendants of the edited individual would carry the edit, okay? And this is actually a natural phenomenon. Apparently there was some element, they call it a P element. Martin probably knows what the element is. But it arose in the 1940s in the Drosophila, the fruit fly in the wild, and apparently it has, following the exact same process just in the last 70 years, driven itself into every single fruit fly in the world. And originally this gene drive idea was proposed as a way through which we could control invasive species. So for example, you could, if you wanted to get rid of all the wild boars in Australia, you could put this female infertility gene into the animal in some way, attach a gene drive mechanism to it, and then use the males to kind of push it into all the pigs. So very quickly you would drive the pig population to extinction. They're doing it also for malaria. There's an experiment, I believe, a real experiment in Brazil. They're putting some sort of infertility gene into Drosophila, or into what are those guys called? Mosquitoes. And they're driving the population to extinction. And similarly, they have done it on an island called Nantucket in the US to get rid of I think it's either mouse or rats that can carry this Lyme disease. So we thought, well, how would gene drives interact with genome editing in livestock breeding context? So it's the same picture as before. Here's our genomic selection. Here's our genome editing if we're editing all our sires for 20 alleles per generation. And then here, the black line is if we do the same thing, but instead of just editing them, we also add this gene drive mechanism. And yes, you get an extra bump in genetic progress by adding this gene drive mechanism.
I mean, is there a simulation in considering also, or will consider also indirect response in other traits? I mean.
So no, it doesn't. I take your point. So you're concerned about pleiotropic effects.
Yes.
So the way I would look at it is, in livestock breeding programs we have one trait. It's called total profitability. And we should be doing GWAS for total profitability, not for the component traits, right? I'm being a little bit argumentative. So you would only edit things that are positive for the total index. You wouldn't edit things that are positive for one trait and highly negative for another trait, I think. Okay, so I talked earlier about the efficiency of turning genetic variation into genetic gain. And here we've got different strategies, and we've compared them for that efficiency. On this axis I've got genetic gain, and on this axis I've got inbreeding. Here I've got genomic selection, then I have genomic selection plus page, where I'm editing for 20 edits all of my 25 sires. Then I've got genomic selection plus page plus gene drive, where again I'm editing all my sires for 20 edits. Then I've got genomic selection plus page, where I'm editing only the top 5 sires for 100 edits. You remember we had that example earlier. And then finally I'm doing genomic selection plus page, where again I'm editing the top 5 sires, but I'm adding this gene drive mechanism. And on this axis we've got, if we look here, we can see the different efficiencies. So let's take genomic selection. It's actually really inefficient compared to the other strategies. So it doesn't make much genetic progress. It stops here. And it doesn't have too much inbreeding, but you don't get a whole lot of gain for that level of inbreeding. If we go to I think the next one is this one, the genomic selection plus page. I've got the labels wrong here.
Is GS and page 5 sires?
Is it? No, it's the red line. Okay, yeah, okay, right. So yeah, okay, yeah. So this is genomic selection plus page for the 5 sires. So it's not that much efficient because it's burning a lot of genetic variation for achieving a relatively smallish amount of genetic gain. But if you go up to this line here, what you see is this is genomic selection plus page, where I'm only editing the top 5 sires, but I add a gene drive mechanism. So in the previous results we saw that editing the top 5 sires does give us more gain, but it also really inflates our inbreeding. But if we add a gene drive mechanism to that, we can get a relative increase in efficiency, because although we're getting quite a bit of inbreeding, we're getting an awful lot more genetic gain for that amount of inbreeding. So actually, the most ethical thing to do according to this metric is to add a gene drive mechanism, which nobody will allow us to do because it's considered unethical, and also to just edit a fraction of the animals, which also people wouldn't allow us to do because that might also be considered unethical. So if you take this genome editing, then perhaps it's time to think about a new breeder's equation. So if you think about what genomic selection has done, so it has decoupled the process of selection from the requirement to have phenotypes. And this was a really important thing. So if we think about agriculture as being 10,000 years old, and if we think about breeding as being an important piece of agriculture all the way back to its foundations, for that entire period, up until there was genomic selection, if you wanted to make a selection decision about an individual, you needed to phenotype that individual or some close relative of that individual. Without that information, you couldn't make a selection decision about that individual. And then genomic selection came along, and it decoupled that. We no longer need to collect a phenotype on that individual or a close relative to make an accurate selection decision about it. And then if you think about genome editing, it kind of goes one step further. It decouples the process of making genetic gain from the process of selection, because to make genetic gain, we don't need to make any more selection. We can just identify alleles and edit those alleles in an unselected set of animals, and we should get some sort of genetic gain as a consequence. So if that was the case, then our old breeder's equation would just be pertaining to the piece due to selection, and we would have to add something due to genome editing. Okay, so now I want to talk a little bit about how we might identify these causal variants to drive this genome editing engine. So you'll remember that over 20 years we edited 314 distinct causal variants, and these were about 3% of all of the 10,000 total causal variants. In any given year, we would need a process that discovers about 15 variants per year. And if we think about how we went around discovering causal variants in the past, we had a single project that would try and identify a single gene. and that was extremely long. It would take 4 or 5 years. It would usually not succeed. It was extremely expensive. So it certainly won't work in this world if we need to identify 15 variants for a quantitative trait per year. So we would like to propose what we're calling an allele testing strategy. And this is a quantitative geneticist approach to molecular biology. And there's nothing new in this except some philosophy. So all of the techniques are well established. The molecular biologists have been using them for years. The only thing that's different is that molecular biologists are in pursuit of molecular truth. Quantitative geneticists are animal science's great approximators. And we are prepared to accept that we do not need to know the truth about something, we just need to be approximately right. And that's the kind of philosophy that we need to adopt, I think, if we are to identify enough targets. So our goal is, let's imagine we're going to edit 100 variants per sire. And the reason we need to edit 100 is that, we want that this 100 contain 15 that are causal and positive, that affect our trait. And the remaining 85, we just want to make sure that they're not deleterious. And the reason they're in there is just to kind of, if we do more editing than we need to, maybe we'll have a good chance that something that we edit has a favorable effect on the trait.
Excuse me, with deleterious, we do not mean unfavorable. It's having a bigger effect.
For deleterious, I mean unfavorable hands.
So just minus is already deleterious. So you
Yeah.
only take the positive ones.
Well, it can be minus but small, because if it's minus and big, and you have 85 minuses and big, that will swamp the 15 that are positive. Right? I guess what it really needs to be is neutral, just not doing something bad, not causing the animal to die or something. And if we could adopt this approach, if anything, this would result in 15 variants being discovered every year, and that would be enough to double the rate of genetic progress in our breeding program. So here's the approach, and as I said, there's nothing new here except the assumption. So allele testing, it's a process to game the odds. So let's make some assumptions. We've got 25 million segregating sites in our pig genome, for example. Let's imagine that 10,000 of these affect our trait. Let's simplify the problem and say, okay, 1,000 of that 10,000 work in some simple additive way. I'm going to leave the other 9,000 to the people that want to worry about complicated things. I'm just going to focus on this 1,000 that I think might be simple. And then I want to have a process that goes in and finds, 15 of these 1,000. And by doing that, I'm prepared to take, a guess at 100 and accept that 85 of those might not be affecting my trait. So what do? We start off, and this is a little bit inspired by a plant breeder's breeding funnel. So in plant breeding, the way it works is you at the top of the breeding program, you just spill in 1 million different genotypes, and then you go through a process of selecting and culling and refining, so that in the end, after let's say 10 stages of selection, you're left with 4 or 5 distinct genotypes that all have passed all the tests and therefore are likely to be good varieties. And this is the same process. Here we spit in 25 million segregating sites. We do some sort of GWAS, and let's say that gives us 1 million variants that kind of survive some significance threshold. We could then follow up with some EQTL study or some comparative genomics, which could whittle this million down to 100,000. We could then do some functional genomics, some of the stuff that Martin was talking about yesterday. We might be able to get it down to a further 10,000. We could then start to do editing of these 10,000. We could get 1,000 of these and put them into real cell lines, do an edit and see do they affect the phenotypes that we could observe in the cell line. That might get us down to, let's say, 500 that we would put into real animals in some sets of cassettes. So we could put 20 alleles into a particular animal and see do those 20 affect the performance of an animal. And this will be, if you like, an in vivo editing testing program. And then finally, this process might leave you with 100 alleles that you would edit in your real breeding program, and you hope that in the end 15 would affect the trait. So as we go through here, we're doing two things. First, we're increasing the proportion of causal variants that remain in our subset. So if we start off, we've got 15 out of 25 million, and in the end we have 15 out of 100, and hopefully, finally, we have all of these 15 affecting our trait. And the second thing we're doing in this process is checking and increasing the probability that the variant is not highly deleterious. So for example, our GWAS might not really tell us whether a particular variant is deleterious. We might be able to tell that from our EQTL, we might be able to tell it too much from our functional genomics, potentially. But we might be able to tell it from some assay in the cell lines, and we for sure would be able to tell it from the editing of real animals. So that's kind of the process. So we've done a little simulation to just quantify how realistic this process is. So we simulated 1 million animals with whole genome sequence information, and they had a trait that was controlled by 10,000 causal variants. And then we performed GWAS on the sequence data. I'm only going to present results for 1% of the genome, and that's a particular window in the middle of a particular chromosome. And we just happen to pick that at random. And that particular window, it doesn't contain the biggest QTN out of these 10,000. The biggest QTN that's in that is the 44th biggest genome-wide. The next one is the 420th biggest genome-wide. The next one is the 574th biggest genome-wide. So I think our results are somewhat biased downwards, because obviously a GWAS that was trying to find a bigger QTN would have a better chance if we had the biggest QTN in our experimental set. So here are the results for that GWAS. So this is the particular window it's the middle of one chromosome basically. It's the 10 centimorgan window in the middle of a chromosome. So the black lines are neutral variants, or the black dots are neutral variants, the red dots are causal variants, and what you see is there are a lot of causal variants down here that don't affect the trait, but there are some ones up here that we are able to find. In fact, we're able to find 4 significant causal variants above this line, where we have 4 red dots up here, but we have 256 false positives, so we have 256 black dots up here. So let's think about what this is in the context of our funnel or our allele testing scheme. So the pre-GWAS ratio, if I just took a random locus and said this one is causal, my success rate at sampling at random would be 1 out of every 1,000. So for every one I would sample that is correct, I would have to sample 1,000 that are wrong. If I do the same thing post the GWAS, I have a success rate of 1 out of 65. So the GWAS is helping us to significantly enrich the set of variants that we have that are significant for causal variants. Right? So if we extrapolate this genome-wide, the GWAS would discover around about 400 causal variants and discover around about 25,000 false positives. So if we put this number in the context, 314 distinct QTN would be edited across our 20 years. So we have already found the QTN that we need to find. We just need to go through and remove these false positives using the rest of the steps in this allele testing process. To achieve a ratio of around about 15 out of 100, we would need the remaining steps to reduce these false positives from 25,000 down to 3,000 and ensure that these 3,000 are not highly deleterious. So I don't know what you guys think of that, but this gives me hope that this is a tractable problem. If without having done this simulation, I would not really believe that we could discover causal variants, but having done it and looked at the numbers, I think we have a chance. So now I want to go quickly and talk about the last piece of this genome editing approach. So Martin told you yesterday that he can run various software packages on sequence data, and he can identify variants that are represented over here that are likely to be deleterious. And then he showed you that this also makes sense because he has an over-representation of deleterious variants in regions of the genome where there's low recombination, and an under-representation in regions regions of the genome where there's high recombination, these areas. And that makes sense because when there's lots of recombination, you have a lot of opportunity to remove these deleterious variants from the genome. Where there's low recombination, they tend to stay there due to linkage drag. And then he showed you that he's able to give every animal in our population a kind of what we might call a load score. So for example here, these guys, on average there's something like each animal carries 1,500 deleterious variants in heterozygous status according to some of these metrics. So then the next question was, could genome editing be used to remove alleles from this population? So previously we were talking about promoting favorable alleles by genome editing, and that gave us a title page. And we were very proud that we could come up with this one because removal of alleles by genome editing would give us the title rage. And because we're working with deleterious load, we could say rage against the deleterious load. And depending on your level of education, that's a reference to, what is it, Martin? Late 20th century culture or early 20th century culture. I didn't know about the early 20th century until Martin told me that. Anyway, I have to get, after my joke, I have to remember what the main message here is. So basically, we can do this genome editing on these stuff that will be deleterious, and we can compare that to some baseline where we're just ignoring the load, Martin, correct? We're just doing nothing with it. We're doing, so that would be the blue line here. Then you can have a process where you avoid the 250 males that have the biggest load score, and you see you get a modest improvement over the baseline. If you do a single edit of a single variant, you get something that is better than both of the baselines. But if you do 5 edits, for example, you can get a significant lift in the fitness of the population by removing 5 deleterious alleles from each animal. So some final remarks. So genome editing could work for quantitative traits. I think the likely next steps will be that in the short term we continue to focus on disease traits, these simple Mendelian diseases. In the medium term, I think we could think about this rage where we would use genome editing to fix up deleterious mutations and potentially lift the fitness of our populations quite a bit. In the long term, I think we should aspire to doing page for quantitative traits because that's where animal breeding works. Animal breeding works with quantitative traits, and if we are to use this technology, we need to make it work in that space. So I'm going to stop there. If there are any questions, So then I'll just say I want to thank you all for the last 3 days. I know everybody has come a long way. You have to listen to us waffle on and talk to ourselves a lot. So thank you very much. It's a real honor to have been here, and we really appreciate you taking the time to come and take part. So thank you.
So John, thank you on this insightful talk, which we appreciate. I appreciate that I told you like one of the millionaires and the most craziest guy in animal genetics. And I appreciate that you are making to provoke ideas and to bring really the things that are bringing difference.
Thank you.
And promoting also new technologies.
Even to Germans.